So let's start. Dear children, in last class we discussed regarding monohybrid cross and some of the numerical problem based on the monohybrid cross. Is it all right? Okay, ji. So today we will start with what conclusion Mendel have drafted based on monohybrid cross. Clear? Is it all right? Okay, ji. So have a look here. So based on a result of monohybrid cross, Mendel formulated three postulates. How many postulates? Postulate you think like the conclusions. How many postulates concluded by Mendel based on the result of monohybrid cross? Three postulates, right? Three law I will talk later. I deliberately am not telling law. Three postulates based on the result of monohybrid cross. First is what? Postulate of paired factor. Second one is postulate of dominance. And third is postulate of segregation of gamete. Segregation of gametes, or it is also called postulate of purity of gamete. Purity of gametes. So these are the three postulates formulated by Mendel. Getting the idea? Is it clear in the mind? Based on his which cross? Monohybrid cross. Voice is not coming. Just a moment. It's not audible for all, or some students are facing the problem coming. So dear student, you check your system. I think most of the students are saying voice is coming. And if you are saying not coming, means there are certain error on your level. There are certain error on your level. Okay. You just go in the settings of the audio. I think after that it will rectify. Okay, ji. So these are the three postulates formulated by Mendel. Postulate of pair factor, postulate of dominance, and postulate of segregation of gamete, or postulate of purity of gamete. And these all postulates are based on which one? A result of which cross? Monohybrid cross. Is it clear? Mendel himself have not given any law. Getting the things or not? Mendel himself have not given any law. So now you can ask a question, then who have given the law, sir? So from the three postulates, based on of which one? Based on monohybrid cross of Mendel, Yeah. It's going on. recordings. Yeah. So from the three postulate based on monohybrid cross of the Mendel, Carl Correns, the name of scientist, Carl Correns, formulated two laws. Getting the things or not? Two laws. Getting the things. The first law is, and that is popularly called laws of Mendel. So you keep in your mind, laws of Mendel is not formulated directly by Mendel. Mendel have given postulates based on monohybrid cross three postulate, and based on three postulate of the Mendel, two law formulated by whom? Carl Correns. Getting the things. So first law is law of dominance. And second law is law of segregation of gamete. Segregation of gamete, or it is also called law of purity of gamete. Law of purity of gametes. Is it clear in the mind? So Mendel have given what? How many postulate? You tell me, you pin your answer. Mendel have given how many postulate based on monohybrid cross? Three postulates. And these all three postulates are based on which cross? Based on which cross? Monohybrid cross. Very good. Is it? Right. And these three postulates are postulates of pair factor, postulate of dominance, postulate of segregation of gamete, or postulate of purity of gamete. These all are based on which cross? Monohybrid cross. And law is formulated by different scientists based on the postulate of Mendel. Name of the scientist was what? Carl Correns. And what Carl Correns did, based on these three postulates, he formulated two laws, and these law is called popularly laws of Mendel. Getting the things or not? One student asking, what is independent assortment? Student, wait. I told in the very beginning, have patience. That postulate, that law is based on dihybrid cross. I am dealing with cross. Based on monohybrid cross. Law of independent assortment is also a law of Mendel. That is based on which one? Dihybrid cross. Getting the idea? So still we don't discussed about which cross? Dihybrid cross. We are discussing about which cross? Monohybrid cross. So these two laws of the Mendel is based on which one? Monohybrid cross. Getting the idea? So we are just going to decode both law. Law of dominant and law of segregation of gamete, how Mendel have formulated the postulate, we are coming on the conclusion of it. So just have a look how Mendel think like this. See the things. Mendel have taken pure line tall plant, pure tall, and cross with the dwarf plant. Dwarf is always pure. Dwarf is always homozygous. Pure means homozygous. What Mendel find? Mendel got in first generation all tall. In first generation, Mendel got what? All tall. Then what Mendel did? These are the result of F1 generation. Then for F2 generation, for F2, Mendel, F2 means second generation, you can say, Mendel perform selfing of F1. So these tall cross with tall. He got tall and dwarf both in the F2 generation. In which ratio? In F2 generation, in 3:1 ratio. These are the result obtained by Mendel. These are the result got by which one? Mendel. Is it clear? Right? Now, you just try to understand how much torturing result is what it was. When parent are dwarf and tall both, only who is coming? Only tall is coming as a progeny. Here Mendel was expecting it may be dwarf will also come. But when he was expecting dwarf will also come, who is not coming? Dwarf is not coming. Got the idea? Is it clear the things or not? And next time Mendel make a selfing of tall only. Selfing of tall only. No parent was dwarf. Male reproductive organ, female reproductive organ, both was which one? Tall, have tall genotype. And in this case, when he was not expecting dwarf, he got the dwarf also. So it is somehow torturing result, na? When expecting dwarf in first generation, because parent was also dwarf, he not got any dwarf. And in F2 generation, when parent was only tall, no parent was dwarf, and children become also dwarf. So it is somehow torturing result or not? Tell me, hurry up. And based on that, Mendel formulated his postulate. What the thought process came in the Mendel mind? Here it is tall. So factor for tallness will there or not? If any plant is tall, you just use your brain. If any plant is tall, factor for tallness will there or not? And who is the factor for tallness? Who is the factor for tallness? Very good, Mahima. Capital T. Factor for tallness is capital T. So it is very obvious, na? If this plant is tall, means who must be there in the genotype? Capital T must be there. Are you agree with me or not agree with me? If a plant is tall, we can say with a very confidence, who must be present there? Capital T. Is it clear, children? It's all right? Very good. Now, if in the next generation, dwarf is coming, who must be present? You know, na, dwarf is a recessive trait and leave the, who must be present? A small t is present. Agree with me or not? A small t is present. Agree? For capital T, for tall, capital T, and for dwarf, small t is present. Okay. Now you can ask a question to me, na? How Mendel told, sir, factors are present in pairs? Mendel told, na? In the law of dominance of the Mendel, this line is also included now. Factors are present in a pair. Okay, Ji. How he told factors are present in a pair? If he not seen the factor, Mendel have no idea about factor. That is the reason for factor Mendel told it is something. Sound is not audible for Lavanya. I think it is a problem of everyone, or you once again asking. You just check your setting, Lavanya. You just go for audio option on your Zoom and you click there and you just check your jack or everything. So I think it may be, it will work. Okay, Ji? So have a look here. So you just come on the conclusion. Here, first generation, he got tall plant. Means capital T must be there. In second generation, he got tall also, dwarf also. So tall means they have capital T. And if dwarf is there, they must have a small t. Then a question came in mind of the Mendel. Ki for F2 generation, who is the parent? F1 is the parent. Agree or not? For F2 generation, who is the parent? F1 is the parent. Is it all right? Yes, G. And for F1, pure line here is the parent. Clear? Is it? Okay. So, if dwarf is coming here, In children, it is not possible if a parent don't have information of dwarfness and it came in dwarfness. In you, only the character which is present in your parent will came, na? Only the character which is present in your parent will came. It not came imported from anywhere else, na? Is it getting the idea or not? Is it getting the idea or not? Right. So Mendel thought like, if this plant is a dwarf, means this F1 parent, the parent of this dwarf must have information for dwarfness. You just try to connect with me. You just try to connect with me. In F1, there must be information for dwarfness. That's only, then only in F2 dwarf will come. Agree or not? Without information for dwarfness in F1, how dwarf will come in F2 generation? Means Mendel definitely forced to think here who must be also present? Information for dwarfness also present. So for information for dwarfness is what? A small t. A small t. Then Mendel thought like it may be factors are present in pair. Because this is a tall plant. This is a tall plant. Getting the idea or not? If a plant, it is a tall plant, it must contain capital T. And their children is a dwarf also, so they must also have which one? Small t. Otherwise, are you in position to give justification of dwarf? If I am saying you justify children dwarf in the F2 generation without thinking about here one small t is also there in the F1, can you say the dwarf expressions in the F2 generation? You are not in position to say. So here information for dwarfness is also there, and this plant is also what? Tall. So here capital T is also there, a small t is also there, and based on that, Mendel make a great conclusion. And what are the great conclusion? Factors are not present alone. Factors are present in pairs. Getting the idea? How the things came in the mind of the Mendels, you got the idea or not? How Mendel forced to think about that, you got the idea or not? He got in F2 generation dwarf plant. He called how it is possible. Why? If a parent is tall, means they must have capital T, and their children have dwarfness, means information for dwarfness is also present in that parent. Means they are tall and they have information for dwarfness. Means how many? One for tallness, another for dwarfness in the next generation. So factors are present in pairs. You understand how Mendel told factors are present in pair? Is it clear? Can I say you just managed to know, dear children, Mendel have no idea about chromosome. One student writing, sir, chromosome present in pair. Mendel don't know anything about chromosome. These are the story before chromosome. Getting the idea, right? Now I am telling you. Now you just try to understand. Now everywhere it is in pair. So I told na, Mendel have taken parent as pure line. So it become capital T, capital T. If I am using green right here, it also capital T, capital T. Here you write a small t, and here capital T. Now it become easy, na? From here capital T will come. From this parent small t will come. This become capital T, small t. And when we make the selfing of this capital T, small t, this tall with this tall, getting the things or not? This tall, we already have seen it. Who will come? Oh. Who will come? Capital T, small t, capital T, small t, two type of gamete will form. Capital T, capital T, capital T, small t, capital T, small t, small t, small t. And these are the phenotypic ratio we are getting. These three are tall, these one are dwarf. Phenotypic ratio came which one? 3:1. You got the idea. Now I am telling you, So how Mendel proved factors are present in pair or occurs in pair? Factors occurs in pair. I hope you got the justification of it. That is proved. It is proved or not? Now I am telling you factor of dominance. Now I am telling you factors of dominance. Got the idea? Why plant with medium height not came, sir? I will talk about the medium height story later. That are the different story. That is the different mechanism. That is the different concept. That is the different inheritance. Got the idea? Have patience. You will learn everything. Learning their children is a process and it occurs step by step. You can't accumulate every information at a single steps, right? So have a look now. So now see what Mendel did. Mendel make a cross between pure tall and pure dwarf plant. But in F1, only who is expressing? Tall is expressing. Dwarf is not expressing. Getting the idea? So Mendel forced to say, here it is capital T is also coming and a small t is also coming. Capital T coming from this parent, a small t coming from this parent. But who is only able to express itself? Capital T or a small t? Who is only able to express itself? Capital T able to express itself. Who is not able to express itself? A small t not able to express itself. So Mendel find whenever I am making a cross between tall and dwarf, one is expressing only and another gets suppressed. So Mendel who told whoever express in first generation is powerful trait. And getting the things or not, whoever expressing in the F1 generation, first generation is which trait? Powerful trait. And it suppress the which one? Another trait. So whoever express in first generation, Mendel told it is a dominant trait, and whoever not express in which generation? F1 generation, Mendel called it recessive trait. Got the idea? Understanding the idea? Is it clear in the mind? So what is dominance according to Mendel? What is dominant according to Mendel? The trait, I also wrote it yesterday, express itself in which generation? In F1 generation is called dominance. So what you are finding? When we make the cross between two different traits of a character, in F1 generation, only one trait is expressing. Both traits are not expressing. So Mendel told the trait when dissimilar factors are there, you see capital T or small t, dissimilar alleles are there. So one allele is powerful, one allele is not that much powerful. So who is powerful allele express itself, who is not powerful not express itself, and Mendel told it is what? Dominant. So how to say about dominant? When we make a cross between dissimilar pair of factor, the pair of factor who express itself in which generation? First generation. F1 generation is called dominant, who suppress, who not express itself in F1 generation is called what? Recessive. So how Mendel proved dominance? How he told dominance? It is not called proof of dominance. It is called, he just anyhow, nothing came in mind of the Mendel other than this. Ki out of two, one is expressing, another is not expressing. So Mendel simply told what? Who is expressing is called dominant. Who is not expressing is called recessive. Is it clear in the mind? So this is about the dominance. Now I am telling you about the proof of segregation of gamete. Proof of segregation of gamete. Two things prove: factors of pair factor. You understand how factors are present in pair, how Mendel told. You also understand who express in F1 is called what? Dominant. Who will not express in F1 is called what? Recessive. Getting the things or not? Okay, Ji. Sir, phenotypic ratio of first generation. Anybody can tell me? Phenotypic ratio of first generation will come what? 1:0. Only one will come, na? F2 generation is 3:1. 1:0. Tall is only coming, na? 1:1 means what? Both are 50-50%. Is it clear? 1:0, 4:0, any 0, you can say that is 100%, na? Okay. Now have a look here, dear children. Now I am going to prove segregation of gamete. What I am going to prove? Segregation of gamete. How Mendel proved the segregation of gamete? For F2 generation, selfing of F1 done. Got the idea? You know, na? This is the F1. And when F1 went for selfing, undergoes in the process of selfing, result of who came? F2 generation came. And in F2 generation, who is coming? In F2 generation, who is coming? Who is also coming? Dwarf is also coming. Who is coming in the F2 generation? Dwarf is also coming. And the ratio of tall and dwarf is what? 3:1. These three are tall and these one are dwarf. So ratio of tall and dwarf is 3:1. Now you tell me, both parent are tall. Children are also dwarf. How it is possible? Whenever, until unless these small t not segregate from capital T and these small t not segregate from capital T, is it possible they will come together? Is it possible they will come together? No, na? Means who is segregating? Gametes are segregating or not segregating? Here, pair of factors are there. So out of this pair or this pair, in this pair capital T is there, small t is there. In this pair also capital T and small t is there. So what you are finding? This small t and this small t came together. It is not possible until and unless these small t not segregate from this capital T, and it is also not possible until and unless when these small t not segregate from this capital T. So expression of dwarf trait in F2 generation become only possible when a small t segregate from capital T. And this segregation of a small t from capital T is called what? Segregation. Separation or segregation is same thing. Yes, dear children. It is called which one? Segregation of gamete. I think I told 2-3 classes back, factors are present as a discrete unit. Here, capital T and small t both are there. Both are present together in a plant, but they never blend. It not blend, na? Blend, it not mix together. So Mendel break the myth of the blending inheritance of its predecessor. Getting the things or not? It is not blending. Okay, Ji? Is it clear? Right? So capital T and small t present as a segregated unit. Whenever small t got the opportunity, they get segregated and they express them. So these small t is present in its purest form. Na? These small t not get contaminated when they are present with whom? Capital T. A small, in this tall plant, who is there? Capital T is also there, small t is also there. But beside of that, a small t is present along with capital T, who not got contaminated. A small t not got contaminated. They not got any character of the capital T. Whenever they got the opportunity to separate, they come with another small t of the another plant, or which one? Same plant, male reproductive organ, better to say, and they just fuse together and just express as a dwarf. And that is the reason law of segregation of gamete is also called law of purity of gamete. Gametes are always pure. Gametes are always pure. They not blend. They not mixed with others. Understand the idea? They remain as a separate and discrete unit. That is told by the Mendel. You got the idea? So capital T and small t are two discrete unit. Discrete means separate unit, and they never mixed. They remain separate always. And whenever they got the opportunity, they get separated. Understand? And this is called segregation of gamete. Is it clear or not? Expression of dwarf trait in F2 generation. This is dwarf trait in F2 generation become only possible when these small t separate from this capital T. And these separation of a small t from capital T, these separation of these small t from capital T, this segregation of a small t from capital T is called segregation of gamete. You got the idea, children? You understood it? So these are, here F2 it is written. Here F1 parent is there, na? Here F2 result is there. These F1 parent and these are F2 result. Clear? It's all right? Now, these are the conclusion of Mendel based on which cross? Mono-hybrid cross. It's over. You understand? Can I give one or two more questions and then we will move on dihybrid cross? Can I give you one more two questions? Okay, Ji, have a look. Question. When we perform the selfing of heterozygous tall pea plant, then what percent of their progeny will be pure line? Answer is 1/4, 2/3, third one is 3/4, fourth one is 1/3. Use your brain and answer it. One option I will change. One option is 1/2. Last option I am changing 1/2. Many are coming as a correct answer. Very good, children. Very good, very good. 50%. You can write, if I wrote in percentage, I am writing here 25%. 2/3, how much is it? 66%, 75%. Getting the things, and it is 50%. Haji. So hurry up. So we are making the selfing of heterozygous tall pea plant. Answer this question. Heterozygous tall. Capital T small t cross with capital T small t. First, every time before answering, we form the gamete. How many type of gamete? Capital T small t, 2 type. And here how many type? Capital T small t, 2 type. Now make the Punnett square. Capital T, capital T, capital T small t, capital T small t, small t, small t. Now you tell me, this is a pure line or not? Capital T, pure line or not? Yes, sir. Very good. Pure line also called homozygous. This is pure line or not? No. This is pure line or not? No. This is pure line or not? Yes. So how many pure line is there? One pure line. It is dominant pure line and it is recessive pure line. Two pure line is there. So 2 by 4 into 100, right? Equal to how much percent? 50%. Answer. You got the idea? You got the idea? Is it? Pure line is the another name of which one? Homozygous. Pure line is the another name of which one? Homozygous. Getting the idea? Understanding the idea? Is it clear in the mind? Okay, Ji. Right. Is done? Then I am changing the question. That question over. I am just making a small change in the question. And these small changes like this. In place of pure line, I am writing monohybrid. Then answer. Percentage view will determine by multiplying by 10, na? 100, na? 2 by 4. How much percent? 50%. Monohybrid. So this is pure line. Many times student confuse pure line. This is pure line. And capital T small t is which one? Monohybrid. Mono means single, hybrid means heterozygous. This is also monohybrid. If it is written like this, we will call it pure line. Don't confuse. And if it is written like this, we will say it monohybrid. You got the idea? So how much monohybrid? 50% is the monohybrid. Monohybrid is also 50%, na? 50% pure line and 50% who is there? Monohybrid is there. Is it clear, dear children? Is it all right? You got the idea? Now we are moving on next. Dihybrid cross means hybridization experiment. Now difficulty level somehow increase. Now difficulty level somehow increase. Got the idea? Okay. Any doubt? No. F3 generation not required. I will tell after dihybrid cross. F3 also, F4 also, F5 also, F6 also. Have patience. Right? Okay, Ji. Genotype in F2 generation, I already told, na? 1:2:1. Law of independent assortment is based on which cross? Dihybrid cross. So as long as you not understand dihybrid cross, how can you understand law of independent assortment? You got the idea? Okay, Ji. Genotype in F1, same 1:0. Phenotype in F1, 1:0. Phenotype in F2, 3:1. Trihybrid there, tetrahybrid there. I will teach you everything. Don't worry. Patiently listen us, you become perfect. You will not make any kind of error. One question is coming, sir, what is meant by genome? Genome is haploid set of chromosome. Haploid set of chromosome, a complete set of chromosome is called genome. Come on the dihybrid. Intentionally, I have taken such area of biology where students somehow struggle, and you will find how much easy is it. If you know a subject in a better manner, in a very professional manner, all things look easy. Some people have phobia of that particular topic, and that is the reason I just started with that particular topic. Sir, how to handle it? Now I'm telling about dihybrid cross. Before dihybrid cross, I want to say you about gamete formation. Gametes formation. Have a look here, dear children. Before dihybrid cross, we require every step gamete formation. If you are not smart in this, you can't move in a single steps in genetics. Genotype in F1, F2. You just tell me genotype in F1, F2. Genotype in F1 what? 1:0. Phenotype 1:0. Because only one type of result came, na? Got the idea? Now come on gamete formation. Suppose that someone asking you a question. Capital T, capital T. Question number 1. Question number 2. Capital T, small t. Question number 3. Small t, small t. Question number 4. Capital A, small a, capital B, capital B. Question number 5. Capital A, capital B, capital B. Question number 6. Capital A, capital B, small b. I am someone going to ask, tell me the type of gametes and name of gamete produced by this. So how many different gamete will produce by this? How many different gametes will produce by this? How many different gamete will produce by this? Examiner can ask. So first I am telling you a formula for types of gamete. This is a formula you keep in your mind. Formula for types of gamete I am writing. What is the formula for types of gamete? Types of gamete equal to 2:n. There is a formula. Type of gamete equal to, there is a formula, and what is the formula? 2:n. Okay, children, are you getting the things? Now you can say, sir, what is the n? Where n equal to number of heterozygous. Where n equal to number of heterozygous trait. Number of heterozygous trait. Got the idea? Is it? Any confusion? Okay, G. So now you tell me, how many type of gamete produced by this? You answer first question. Type of gamete produced by this. Put the formula. What is the formula? 2:n. How many heterozygous is there? How many heterozygous is there? In first question, how many heterozygous is there? 0. Very good. So what should you will write in place of n? 2:0. Value of 2:0? 1. 2:0 equal to 1. A:0 equal to 1. Any value, any power is 0, its value is what? If it is 5:0, 1. 2:0, 1. Getting the idea or not? It's all clear in the mind? Now come on this second one, type of gamete. What is the formula? 2:n. Here, what is the value of heterozygous? How many heterozygous is there? How many heterozygous is there? This is a trait, na? Factors are present in pair. This is one pair, na? And both are dissimilar or similar? Dissimilar alleles are there. So heterozygous You know how to identify heterozygous. I think that much you know now, na? You know now, na? Homozygous, heterozygous. Or anyone have any confusion? Anyone have any confusion in homozygous, heterozygous? Anyone have any confusion? Any children, if they are, yes, right. Yes, sir. One student came. So look, when I wrote capital T, a small t, this is homozygous. So when in a pair, both allele are of same type means either both are in capital letter or both are in a small letter. We say it what? Homozygous. Homozygous. If I wrote capital T, small t, here one in capital letter, another in a small letter, both are T itself, but one in capital letter, one in a small letter. So here it will write heterozygous. So you identify how to know homozygous, heterozygous. I hope no one now get stuck now. No one get stuck now. Homozygous, heterozygous identification. Now tell me, it is heterozygous or homozygous? Heterozygous. How many heterozygous? One heterozygous. Agree or not? One heterozygous. What is the formula? 2 is to the power n. 2 is to the power in place of n should we write 1? So how many type of gamete will produce? 2 type of gamete. 2 type of gamete. Here, how many type of gamete will produce? 2 is to the power n. 2 is to the power, what should I write? Homozygous or heterozygous? Homozygous. So we write 0. How many type of gamete will come? 1. Now here tell me children. 2 character are there. One character is this one, controlled by capital A, small a. Another controlled by capital B. Agree or na? 2 pair are there na? Here not only 1 pair is there, 2 pair. So 2 pair, 2 different character. So here this is heterozygous, this is homozygous. How many type of gamete will come? 2 is to the power n, 2 is to the power 1 equal to 2. Anybody have any problem in 4th question? Type of gamete, you can tell me, sir, I have problem. I will give you the rectification. Anyone have any doubt in the 4th one? I have, sir. Tell me, dear children. 4th one I am writing. Capital A, small a, capital B. I am asking how many type of gamete will produce. What the formula we have given? 2 is to the power n, where n equal to number of heterozygous. Tell me, is it heterozygous? 1 pair, 2 pair. This pair is heterozygous or homozygous? First pair is homozygous or heterozygous? Hetero. Second pair is homo or hetero? Homo. So how many heterozygous? How many heterozygous? 1. Agree or not? So what the formula? 2 is to the power n. We write 2 is to the power 1. Can we write here like this or not? And value of 2 is to the power n equal to 2. So 2 type of gamete. Clear? Clear? Over? Is it all right? Now, we are going to move ahead. This one. Tell me this one, 5th one. How many type of gamete will produce? Once again, no heterozygous. 2 is to the power 0. 2 is to the power n. Getting the things or not? And value of n equal to which one? 2 is to the power 0 equal to 1. Why 2 is to the power 0? It is not heterozygous. It is not heterozygous. Both are homozygous. And in that case, first character is also heterozygous. Second character is also heterozygous. So 2 is heterozygous. 2 is to the power n. 2 is to the power 2 equal to 4. You got the idea, children? So can I expect you are confident in this area? You will never make a mistake. You will never make a mistake. Done, Ji? Okay, all right. Few more questions. Okay, look. Now, number of produce, the different number of gamete produced by capital A, small a, capital B, small c, capital D, capital E, small e. Tell me the number of gamete produced by it. Now question become big, but easy. Big but easy. So how many character? You just have to circle one character, two character, third pair, fourth pair, fifth pair. 5 different genes are there. Okay, Ji, how many are heterozygous? How many are heterozygous? First one is heterozygous. Yes. Second one is not. Third one is small c, not. Fourth one capital D, not. Fifth one capital E, small e, yes. How many heterozygous? How many heterozygous? 2 heterozygous. So what will happen? 2 is to the power 2 equal to 4. You got, dear children? You understand it? You understand it? Clear, clear, clear. Very good. Done. Can I give one more question? Can I give one more question? Okay, okay, okay. Small a, capital B, small b, capital C, capital D, small e, small e, capital F, small f. How many type of gamete will produce? So first you count heterozygous. So look, once again, this 1 character, 2 character, 3 character, 4 character, 5 character, 6 character. 6 different genes are there. Okay, so is it homo or hetero? Homo. I am taking another color. Homo, hetero, homo, hetero, homo, hetero. How many are hetero? 3 are heterozygous, na? So what happened? 2 is to the power n, 2 is to a value of n is 3, 2 is to the power 3 equal to 8. Clear? Understood? Okay, Ji, all done. So I hope you got perfection here. You will never make a mistake in any question, anyone going to ask you related to which one? Gamete formation. Can I expect, dear children? Can I expect? Yes. Yes. Now you can ask a question, na, ki sir, how to know what is the name of gamete? Type of gamete, you told us the formula, sir. But you also tell me, na, what are the name of gamete produced? So how to know the name of gamete? This is a formula for type of gamete. Now I am telling what is the tricks for name of gamete. I am telling a tricks, name of gamete. So for name of gamete, there is a formula. Suppose that someone going to ask you, tell me the name of gamete produced by it. Tell me the name of gamete produced by it. Here, how many gamete will produce? Tell me the type of gamete first. Tell me the type of gamete. Type of gamete is 2. Types of gametes. In this case, first case is how much? 2. In second case, how many type of gamete? In second case, how many type of gamete? Capital A, capital B, small b, capital C, small c. Capital C, small c. All 3 are heterozygous. So in this case, type of gamete is 8. Now if someone asks you, children, tell me what are the name of these 2 gamete? Here 2 type of gamete produced, na? Here 8 type of gamete produced. What are the name of 8 gametes? Okay. So now I am telling the formula for that. How to give the name of gamete. Very. So name of gamete. Someone written correct, someone written incorrect in answer from your side. Name of gametes. Don't worry. You will never make a mistake after this 10 minutes. You will find confident and perfect in this area. You will never make a mistake. That is my confidence. Okay. Have a look here. See the things. See the things. So name of gamete, formula, tricks. We use a method called fork line method. What the method we use to write the name of gamete? Fork line method. There is a method called fork line method. Put fork before heterozygous. And put line before homozygous. So simply keep these things in mind. If a heterozygous is there, put a fork before that. If heterozygous is there, put a fork before that. And if a homozygous, put a fork before heterozygous. And if homozygous is there, put a line before homozygous. And then read it. You can say, sir, use it. How can you use this tricks or formula? Getting the things or not? So I am using it. Can I use this formula for first question? How many different type of gamete you already wrote? That is 2 as a answer. Now, what are the name of these 2 gamete? I am just telling you. Have a look. See the things. So here in question, who is there? Capital A, small a, capital B, small, capital B, capital B. This is the question. And we are asking, tell me the name of gamete produced by it. So you tell me, dear children, this is heterozygous or homozygous? Tell me. Heterozygous. What I told? Put what? Fork before heterozygous. So I am putting a fork like this. And I am writing here capital A, small a like this. You got the idea? How I am writing capital A, small a, you got the idea? Understood? Is it clear? Now, it is hetero or homo? Homo. Very good. Now put line before it. Capital B, line, capital B. How to put the line? You got the idea? Now read it. Capital A, capital B, small a, capital B. This is one type of gamete. This is another type of gamete. Total how many type of gamete? 2 type of gamete. Clear? Clear? I'm giving you 3, 4, 5 question more. So you got the perfection in your practice. I am committing you now. After this class, no one can trap you at any level, at NEET level or any exam level. No one can trap you. Okay, Ji, can we move ahead? Can we go for another question? Can I frame 5, 6 question? Okay, I am framing 5, 6 question. Like first, so you got a perfection. First question. Everywhere you have to say the type of gamete. After doing that much question, definitely we got the perfection. And you will say, sir, you ask anything, I will never make a mistake. Always develop confidence in you. Got the idea? So first question we are going to solve. Question is this. How many type of gamete? This is the question. In front of us. We are asking type of gamete. So, what we put here? Homo or hetero? Tell me. Homo. Agree or not agree? So, before homo, we put fork or line? We put line. And we write what? Capital A. In line, what we write? capital A. Agree or not agree? Yes, G, agree. Now, second character, homo or hetero? Homo. What we put before it? Line. And we write capital B. Read it simply. Capital A, capital B. How many type of gamete? One type. Formula also saying one type, na? 2 is to the power 1 equal to 2 is to the power 0 equal to 1. One type of gamete came or not came? It will give how many type of gamete? Capital A, capital A, capital B give how many type of gamete? It become 2 is to the power 0, na? And 2 is to the power 0 equal to 1 type. And this is the one type of gamete. This is the one type of gamete. Name of one type of gamete. Now, come on second. Second here is, I am writing like this. A, capital B, capital B. What is the type of gamete? Type of gamete equal to 2 is to the power n. Here it is a heterozygous. So 2 is to the power 1 equal to 2. Two type of gamete will produce. What is the name of two gamete? For writing name, I told you a formula, fork and line. So here it is homozygous or heterozygous? Homozygous. We put line and we write small a. Here it is homozygous or heterozygous? Heterozygous. We put a fork like this and we will write capital B, small b. Coolly you look. And what it become? Small a, capital B, small a, small b. Small a, capital B, small a, small b. This is one type. This is second type. You got the idea? Understand the idea? Clear? Okay, Ji. Now we are practicing the third one. Third one I am practicing here. So it is first, it is second, and here we are going to discuss about the third one. So capital A, small a, capital B, small b. How many type of gamete will come? 2 is to the power heterozygous, heterozygous. And 2 is to the power 2 equal to 4. Now what are the name of 4 gamete? You will ask, na, sir, tell me the name of the 4 gamete. So it is hetero or homo, dear children? Hetero. We will put fork or line? Fork. So fork we will put like this. Capital A, small a. This is hetero or homo? Hetero. We will put fork. We will put fork here also, here also. And we will write capital B, small b, capital B, small b. Like this. Okay. Now read it. Capital A, capital B, capital A, small b, a small a, capital B, a small a, small b. How many type? 1 type, 2 type, 3 type, 4 type. It is proved or not proved? It is proved or not proved? Proved, na? Is it clear? Now we are going for this one. You do it. You do by yourself. 4th you do, 5th you do, 6th you do. Otherwise one I am doing. You tell me which should I do out of these 3. To whom you will say I will do? 4th, 5th or 6th. What do you expect from me? Most of the student writings are do the 4th. Okay, I am doing the 4th. I am doing the 4th question. It is capital A, capital B, small b, capital C, capital C. It is it is only one heterozygous. So 2 is to the power n, and here you will find 2 is to the power n value is 1 equal to 2 type of gamete will produce. How 2 type of gamete look? It is hetero or homo? Homo. We will put line and we will write capital A. It is hetero or homo? Hetero. We will keep fork. Capital B, small b. We will write like this. Before hetero, we will write like this. It is hetero or homo? Homo. We will put line. We will write capital C, capital C. We will read it. Capital A, capital B, capital C, capital A, small b, capital C. How many type of gamete came? 2 type of gamete came. Are you getting the idea? Understanding the idea? How fourth done? Is it clear, children? Is it all right? Any problem? Any doubt? Now your turn. Now it is your turn. Try to do fifth and sixth by yourself first and write the answer. After that, I will solve fifth and sixth both. Gamete not occur in pair. Factors occur in pair. In somatic cell, not in gamete. In gamete, in monohybrid, I also told out of capital T, this capital T will come in gamete. So how gamete will come in pair? This is the gamete. Na? Gamete never occur in pair. Good one. Many students doing very good. Now I am doing fifth, and you check how many people answer are matching with my answer. So I am doing fifth one, and your duty to you just match your answer with my answer. This is the question. Capital A, capital B, small b, capital D, small d. Here type of gamete, how many heterozygous? This is heterozygous, this is heterozygous. 2 is to the power n, 2 is to the power 2 equal to 4 type of gamete will come. What are the name of all these 4 gamete? So we hope go for fork line method, hetero or homo, homo. We will write line, we will write capital A. Next is hetero or homo, next is hetero. We will put fork, capital B, small b will write like this. Next is hetero or homo, hetero. We will put fork here also and this place also. And we will write capital D, small d, capital D, small d like this. Now what become? All done, all over na? Now capital A, capital B, capital D, read it. Na? Capital A, capital B, capital D. Now read capital A, capital B, small d. Capital A, capital B, small d. Next capital A, small b, capital D. Capital A, small b, capital D. Next capital A, small b, capital D. 4 type of gamete produced. 1 type, 2 type, 3 type, 4 type. Your answer is matching with my answer or not matching? Matching. Okay, well done. Well done. Well done. Okay, Ji? Right. Now I am telling you the which one? 6th one. Okay, children? You got the confidence in this area or not? You got the confidence? Slowly you are getting confidence in monohybrid cross now. Okay? Right. Okay, now the last question, 6th question. So 6th question was what? Capital A, small a, capital A, small a, capital B, small b, capital C, small c. All are heterozygous. 1, 2, 3 heterozygous. So 2 is to the power n, 2 is to the power 3 equal to 8. Now what are the name of 8 gamete? So all are hetero. So I'm writing hetero, capital A, small a. Hetero, capital B, small b, capital B, small b. In one line, one character will come only. Not like here capital B, small b and here capital C, small c. Don't do that blunder. Here, everywhere we put fork like this. Capital C, small c, capital C, small c, capital C, small c, capital C, small c. Now we have to read it. How to read? Capital A, capital B, capital C. Capital A, capital B, capital C. Capital A, capital B, small c. Capital A, small b, capital C. Capital A, small b, small c. Everything from capital A over. Now small a, capital B, capital C. A small a, capital B, small c. A small b, capital C. A small b, small c. How many are there? How many are there? 8 are there, na? 8 are there. Done. Over. So how to handle? Is it very easy or not? Tell me. But if you are not done methodically, many students struggle. Many people came to us in long term even and they struggle to do this. After doing their intermediate, after passing first year and second year, many students came like that. They are struggling in this. Right? So it is easy, na? So if you are methodically doing everything, dear children, the NEET exam look like a easy cake. Trust me. Getting the things or not? It look like, oh, what is the standard of the question? I just crack it. Getting the things or not? In the very first attempt with a great score. Is it or not? So have a faith on you. One thing is very clear. As you started early, you people are sincere. Many of your friends are still playing. Getting the things or not? But you started. That is going to show your level of sincereness. Maintain it, dear children. Maintain your sincereness. Everything become happy, rocking. Time completed. Yeah, I'm giving you the QR code. Time is over. Okay, have a good day to all of you. Thank you very much, children. Okay, see you soon. Bye-bye. Okay, definitely we are also eagerly waiting for you in the campus. Definitely, children. See you. Bye-bye. Bye. Thank you. Thank you very much for your high words. Thank you, dear children. Bye-bye. Bye-bye. Bye-bye.